The overall goal of this procedure is to monitor synaptic transmission through the giant fiber pathway by recording postsynaptic potentials from a dorsal longitudinal muscle, or DLM, and from a tergotrochanteral muscle, or TTM, following direct stimulation of the giant fiber interneurons. This is accomplished by first properly securing an anesthetized fly in a dish containing a dab of wax. The next step of the procedure is to insert stimulating, ground, and recording electrodes into the fly. The final step of the procedure is to stimulate the brain and record responses from the jump and flight muscles. Ultimately, results can be obtained that show changes in the functional properties of the giant fiber pathway as a function of a genotype, age, temperature, etc., by recording the amount of time between the stimulus and the response, and by counting the number of successful responses following a high-frequency train of stimuli.
This method can help some of the key questions in the field of neurophysiology and help examine the functional properties of neuronal pathways. This technique can be employed either as an introductory experiment that is followed by genetic or molecular analysis, or as a way to assess physiological and functional properties in animals carrying a known mutation.
Visual demonstration of this method is critical, as the preparation and impairment steps take some time to learn. Because flies are comparatively small, there's a certain level of steadiness and coordination that needs to be acquired before experiments can be done correctly.